Do you know the algorithm or reason behind the flower pattern, zebra stripes, leopard spots, and spiral seashells? Welcome to your own channel ADP for Learning. I am your own learning partner Ashish. Today we will be learning about the algorithms of beauty, that is the mathematical genetics of various patterns that are found in various plants and animals. Nature is full of striking patterns. Zebra stripes, leopard spots, and spiral seashells are just a few of the endless examples. Have you ever wondered how these patterns form? It's a question that's fascinated researchers for a long time. The search for the answer spans several fields, including biology, chemistry, and mathematics. The answer to these biological patterns lies in Turing patterns. In 1952, the mathematician Alan Turing proposed a model for how biological patterns form. Using mathematical equations, as shown in the figure, he described how substances spread through space and react with one another to create patterns. He called the substances morphogens, an intentionally vague term. In the context of pattern formation, it can refer to chemicals, genes, proteins, or even groups of cells. The important thing is that one morphogen, an activator, makes a change that leads to a trait. A second morphogen, an inhibitor, stops the change. The activator increases its own activity, and it turns on the inhibitor too. The activator and inhibitor diffuse, or spread, at different speeds. Turing's model could explain the spots on a ladybug, for example. Let's say A is an activating morphogen. It causes black pigment. I is an inhibitor that prevents the trait. Where A is produced, it makes more of itself. It diffuses slowly, so a lot of A builds up in a small area. A also turns on I, but I diffuses quickly. There's a small amount overlapping with A, but most moves further away. As a result, A is high and I is low in a small area. Black pigment is made. Further away, A is low, but there's plenty of I. No black pigment is made. Turing's work is now known as the reaction-diffusion model. An important point is that it only requires two morphogens to establish a periodic pattern. In computer simulations, changing a small number of variables causes a dazzling variety of patterns. Many resemble those found in nature. The spots on a ladybug can be considered as a good example of reaction-diffusion equations. Variables include the amount of morphogen present, its diffusion rate, and how strongly the morphogens affect one another. As shown in the figure, many patterns can be modeled with the equations. The reaction-diffusion model can also be seen in plants in the form of petal spots. Many other real-life patterns could form from the reaction-diffusion model. Proving they actually do is more difficult. The hunt's been in progress for a long time, and verified examples of Turing patterns are still scarce. The few identified so far include zebrafish stripes, chick feather buds, mouse hair follicles, and flower petal spots. Two key pieces of evidence are needed to prove a pattern formed by reaction diffusion. They are the identity of the activator and the identity of the inhibitor. In the monkey flower, also known as Mamulus lewisii, researchers first solved this puzzle. Then, they tested if the activator-inhibitor pair they found fit other predictions of the model. Monkey flowers have nectar guides, spots of dark pink anthocyanin pigment on a yellow background, that attract pollinators. The guides form from the activator-inhibitor pair nectar guide anthocyanin, negin, and red tongue, artio. Both negin and artio are transcription factors, proteins whose job is to turn gene activity up or down. Negin and artio fit the reaction-diffusion model well. Negin turns on genes needed to make anthocyanin pigment. It also activates itself in artio. Once on, artio inhibits negin by competing for a specific partner protein they both need to do their jobs. Artio diffuses from where it's made into neighboring cells, restricting negin's activity. As shown in the figure, the pattern of anthocyanin spots on monkey flower petals is a real-life example of the reaction-diffusion model. The dissected monkey flower petals shown in the figure are from genetic experiments to test and expand the model. As on the left of the figure, Both activator and inhibitor are present, resulting in normal spots. While in the middle figure, no activator is present, so it leads to no spots. On the right side of the figure, there is no inhibitor that resulted in expanded spots. As shown in the figure, the analogy of a traffic light can help us in better understand the activator-inhibitor pair concept of Negan and RTO transcription factors that fit in the predictions made by the reaction-diffusion model. It is important to note that these patterns also influence the pollinator's choice. Flower patterns don't just look nice, they're also really important. Patterns help attract pollinators to a flower. The more pollinators visit a flower, the more likely it is to reproduce. In monkey flowers, petal patterns affect pollinator choice. Mimulus luteus is a species of monkey flower that grows in the Andes Mountains. Even within a species, patterns differ. Some flowers have broad patterns and are favored by hummingbirds. Others have narrow patterns and are favored by bees. Because patterns influence pollinator choice, it's possible that reaction diffusion played a role in flower evolution. Small changes in reaction diffusion networks have a big impact on patterns. This could be part of the reason we see so many diverse flower patterns today. The figure clearly shows examples of patterns in different species of monkey flower. Mimulus rupicola on the top left, Mimulus variegatus on the top right, Mimulus luteus on the bottom left, and Mimulus pictus on the bottom right. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.